Good morning, everyone. Welcome. In this talk, we are going to talk about infrastructure challenges, machine learning in the cloud as a future-oriented solution. Let's get started with introducing speaker and agenda for this talk. My name is Ondrej Klempir, and I'm Senior Community Engagement Scientist at DNA Nexus, mostly focused on imaging data and UK biobank data. In this talk, we will be covering the following. We will be introducing DNA Nexus. We are going to talk about biobank as a large-scale data set. We are going to talk about ways and mental models of working on the cloud. And then we will show you two machine learning scenarios on the cloud. First, image augmentation using MONAI and training models on image-derived phenotypes. As I mentioned, I work for DNA Nexus. DNA Nexus is a company providing a clinical genomics data platform that accelerates innovation and collaboration to improve human health. And we have many customers. And one example of such customer is UK Biobank. And we are strategic technology partner of UK Biobank, covering platform, which is called UK Biobank Research Analysis Platform. All right, let's talk about problem statement. One of the main challenges in neuroimaging-based machine learning and artificial intelligence is the lack of large datasets with rich phenotypic information available for machine learning applications. And it's general knowledge that machine learning models work best with lots of high-quality data. There is another issue, which is related to storing all of the big data in a centralized location and making this data easy for large teams to perform analysis and collaborate. The UK Biobank provides a solution to one of the issues, since they collected various biomedical data for more than 500,000 people across the UK. And more importantly, data contains information about the international classification of diseases and also results of cognitive tests, which can be used in aging research. With that being said, on this slide and in the next part, let's focus more on UK Biobank Research Analysis Platform powered by DNA Nexus. UK Biobank Research Analysis Platform is a secure research platform hosted on AWS London region. It's bringing the researcher to the data, making data access and compute more widely accessible. UKB RAP allows secure and fast analysis of UKB data, and importantly, brain data is key part of imaging, with over 50,000 participants scanned to date. So what are the options for working with UKB data on the RAP and how you can create machine learning models on the cloud? On DNA Nexus offers many tools already available for the analysis. One of them is called Browser, and we will be showing one demo during this talk. Then you can typically work with workflows, either write it via WDL or NiPipe, or you can create your own machine learning models in the Jupyter Lab environment. Basically, you can install software packages in these different environments and bring your own tools on the platform to the cloud. So let's talk about cloud-based analysis on the cloud and on DNA Nexus and talk about some mental models when working on the cloud. We will be working in the following flow. First, we would need to request worker, which is called instance, and also provide some environment. In our case, that would be machine learning environment with all the tools available. This means that Cloud Worker is launched and managed by DNA Nexus. Then, as an important step, we would need to transfer scripts as well as data from some permanent storage to the worker. Then we can do our computation. Let's say we would need to unzip our files with our images and then execute our algorithms or train the models or apply models to the new data. And as a last important step, we would need to transfer all the results and models back to the permanent storage so we can create model versions or create snapshot with the environments. Let's consider one example that we are running machine learning models with graphical processing units on UKB RAP under Jupyter Lab environment. We can work in two versions. We can work with CPUs. This is typically sequential series processing with multiple cores, good for many types of computations. But when we work with machine learning and do some really computational intensive tasks, we can use graphical processing units, which make the computation much faster. So as one example of such infrastructure, we can select appropriate instance type with graphical processing units in the suffix. And then once our machine is ready, we can check if and confirm if GPU is installed. We can find many tools already available on the cloud. Here on this slide, we can see examples highlighted in red color for image processing and for machine learning frameworks. Examples might be TensorFlow, PyTorch, Scikit-learn, or let's say Keras, and these tools are compatible with graphical processing units. So now we all understand the basic principles of cloud computing, so we can deep dive into machine learning scenarios. Let's first talk about image augmentation using MONAI deep learning. MONAI is a PyTorch-based framework for deep learning in healthcare imaging. It's a fast way to build deep learning pipelines. It can be easily installed in the cloud. It can run on GPUs. And what are the typical machine learning MONAI tasks? It's a classification, segmentation, data labeling, and data augmentation. And what is augmentation as a task? For specific cohorts like dementia or Parkinson's disease, we have a finite amount of data, so to avoid overfitting, we can apply random transforms to modify our data. And this can provide an easier, faster way to augment data compared to generating synthetic data. Here, let's concentrate on some MONAI transformations applied to UKB MRI data. You can basically compose multiple transformations together and tune the different transformation parameters to obtain a new derived images. You can apply operations such as translate, rotate, round, affine, scale intensity, and many, many more. Second machine learning scenario will be about training models on image-derived phenotypes. In this scenario, we are analyzing image-derived phenotypes on BLE Nexus cohort browser. UKB imaging data were already preprocessed by specific neuroimaging pipelines, and this data was ingested into the form of database. We can explore this database in the tool which is called Cohort Browser. This might be super interesting for genomic scientists or disease-specific researchers too. We can basically create cohorts based on tissue volume quantification. This can then be fed into the machine learning algorithms or genome-wide association studies. On this slide, we can see a list of specific volumes or extracted data from the specific brain regions on the left side. And based on these fields, we can then create cohorts and select specific participants with specifying filter. Based on that, we can then create some simple plots and explore data visually. For example, create scatter plot and see some correlations in the data. UK Biobank dataset is not only about healthy people. The UK Biobank contains a series of diagnostics data classified following the international classification of diseases. So we can focus on specific category, which might be dementia in Alzheimer's disease, and study the specific cohort of participants. As part of our machine learning experiments on the cloud, we focused on the following. We tried to train a model to predict age, explained by several brain volume features. Our model was trained using only neuroimaging process data. This data was in a tabular form. We programmatically defined cohorts in JupyterLab. We focused on 3K participants in the selected cohort. And we evaluated our models using 10-fold cross-validation and by the following metrics: Pearson correlation coefficient, R-squared, and mean absolute error. We used lasso model from scikit-learn. And for reproducibility reasons, we then saved our notebook plus model plus environment to the permanent project in the cloud. Let's look at some data examples and let's get started with table representation. We were able to obtain correlations of individual neuroimaging features with age. And we were also able to obtain a couple of plots showing the results of our model. On the left side, we can see principal component analysis, and on the right side, we can see lasso regression to predict age based on gray volumes. Each color represents one cross-validation fault. And just to wrap up the things and do some summary, in this presentation, we talked about infrastructure challenges and how to do machine learning on the cloud. And we specifically talked about MONAI and how MONAI transforms provide an easier, faster way to augment data compared to generating synthetic data. We were also talking about how to deploy models in UKB RAP powered by DNA Nexus Cloud and how this can lead to better model versioning and reproducibility. And with that, actually, how raw data can be annotated with the results of machine learning analysis. So this may accelerate the work of clinicians and translational researchers. Last but not least, I would like to say thank you to my DNA Nexus colleagues, and presented materials were carried out using UK Biobank application number 46926. Thank you very much for your attention.